So welcome. My name is Kelsey Hansen. I am an educator at the DNA Learning Center in Brooklyn at our new-ish New York City location. And it's my pleasure to join you at this Meet a Scientist series and to welcome our first scientist from REGENERON, Zachary Oberholtzer. My first thing I want to mention is that we have just started, we've just listed our summer camps for this year's season. So I'm going to actually share my screen real quick because I want to let you know about some of the cool things that you can do at the DNA Learning Center if you're not already familiar. So I just want to share really briefly that we currently have a few Saturday DNA events right now. Just quick little labs, mostly for the middle school age at all three of our locations. So the Brooklyn one has now been rescheduled to February 11th, but we've got labs at Cold Spring Harbor and also at Sleepy Hollow on the REGENERON campus that are happening on Saturdays. Just a quick little activity for any middle school students. We also are doing a few school break biology sessions here in Brooklyn. So the Wednesday of school break, we'll have a little bit longer labs for middle school and high school students. If you're not traveling for the fall or April, or the February or April breaks, feel free to bring your kids by. It'll be a fun time. And then the final thing that I want to mention before we jump in is that our summer 2023 summer camp registration just opened. So we will have virtual camps, but also a lot of in-person programming at the Dolan DNA Learning Center, the original on Long Island, at our Learning Center in Brooklyn, and also in our DNA Learning Center at REGENERON in Sleepy Hollow. So if you go to our summer camp page, you can click on your desired location. You can see a little bit of information about the site, and then you can use this guide to figure out when you have availability and what types of labs you would be interested in. So we have summer camps for everywhere from rising sixth grade through seniors in high school and all sorts of topics, mostly related to DNA, but we have a couple enzymes sneaking in there. So if you want to learn more about our summer camps, you can start a parent profile and register your kids to come see us. I'm going to stop there. Oh, actually, I want to share one more thing. Because you're here for the Meet the Scientist, if you want to learn more, we have a few great speakers from REGENERON coming up later this spring as well. So Terrence Turner and Stephanie Woods will both also present their journey in science and what they do at REGENERON as well. And if you want to see some of our older past events, you can go to our YouTube page, or also if you go to our Meet a Scientist page here, you can find links to the videos from those talks. So without further ado, I'd like to thank Zachary Oberholtzer for being here with us today, and he's going to tell you a little bit about his journey and also the role of biochemical and biophysical characterization in drug development.
Okay, thanks, Kelsey. My name is Zach Oberholtzer. I'm a research specialist at REGENERON Pharmaceuticals. And today I'm going to talk a little bit about my career journey, as well as what my department does, what me and my colleagues do in the drug development process. So just to get started, why would you want to go into science, or why would you want to get a scientific education? Well, STEM and science offers a versatile tool set to tackle the world's problems. Not only do you get a lot of background knowledge in a basic understanding of how the world works with basic science and basic math classes, it also teaches you how to think critically and develop logical thinking skills and make argumentations from data. But also, contrary to what might be a popular stereotype, it also teaches you important communication skills. In science, you have to be able to communicate your findings, otherwise your research is not going to have the impact that it should. In addition to preparing you for a career directly in scientific fields, a STEM background can also be useful for a launching point into many other types of careers. Here I have just shown a few examples, by no means a definitive list, and a lot of these are scientific adjacent, but there are things further afield listed on here, things like marketing, things like business development. So a STEM career doesn't just limit you to traditional research kind of based careers. So just a little bit of background about myself. I'm born and raised in Harrisburg, Pennsylvania. And when I was in high school, I spent a lot of time, or I took a bunch of AP classes, which is by no means a prerequisite for a career in STEM, but it was helpful for when I went to college, helping me have more flexibility in my course curriculum when I had to take classes, what classes I was able to take. I also was on the wrestling team, and I spent my summers working concessions at Hershey Park, which taught me the value of hard work as well as being nice to service personnel. I also spent a lot of time reading fantasy books, which is something I still enjoy doing today. So at Bucknell University, where I did my undergrad, I studied chemical engineering. And a few of the reasons I went into chemical engineering, first, engineering and science degrees are pretty easy to transfer out of, but harder to transfer into. And the reason for that is usually you have very sequenced course structures. So you have to take, for example, your Calculus 1 course, then your Calculus 2 course, then your Calculus 3 course. And it makes it harder to move things around and have flexibility in your schedule because you have to take things in a certain order and things are only offered at a certain time of year. But if you start in engineering or science, since a lot of those early classes in basic math and basic fundamental science, there's a lot of overlap between them in the requisite courses you have to take. So starting there, it makes it easy to transfer within different scientific degrees, but also easier usually to transfer out is usually a lot of those basic science classes you can use to take up other requirements in your new degree. Chemical engineering is a pretty versatile field. It covers everything from medical research all the way through paint manufacturing. And when I originally started, I thought I wanted to do medical research or something that was involved with helping people, but I was early in my career. I didn't really know exactly what chemical engineering entailed, wasn't quite sure I'd like it. So I liked the idea of a degree that would allow me to do a lot of different things. And then I also had a good AP chemistry teacher in high school and just enjoyed chemistry. So chemical engineering seemed like a natural fit for me. So a lot of people wonder when they go to college what degree they should pursue. And in undergraduate, to a certain extent, it doesn't matter. As I said, there's a lot of overlap in kind of the early courses you have to take. Usually you have to take the same math courses, no matter what kind of STEM field you're going into. And while there is some specificity, for example, if you wanted to do something in computers, you'd probably want to get a degree in computer science or computer engineering. There is a lot of overlap in, again, early courses, but also in a lot of the soft skills you develop. There's also demand for kind of cross-functionality in a lot of different fields. For example, with a computer science degree, that doesn't mean you couldn't go into a field like biology later. In biology, there's a lot of demand for things like computational scientists and people with computational backgrounds. So having that degree in computer science isn't going to limit you to just working in that limited field. So I want to say that it's not that it's not important what degree you choose, but it's not going to be definitive of what you can do later in your career, especially if you're willing to work hard. I also want to advise not to waste your electives. Most people only spend one time going through undergraduate, so it's your one chance to take courses that are maybe not going to be necessarily directly tied to your eventual field of work, but courses that interest you. For example, myself, one of my proudest accomplishments in undergrad was I got into the English Honor Society based on a bunch of creative writing poetry classes I took. Those poetry classes actually were also valuable for teaching me communication skills and valuable for my scientific education because it really allowed me to learn how to really think about what words I was using and how my sentences and sentence structure was communicating the ideas I was trying to communicate. So a few other things I'd advise is use internships and on-campus research opportunities to explore fields. Go try things, try a lot of different things, see what you like, see what you enjoy, and think about whether it's something you would want to make a career of. And also just remember that careers are long. It's not a race. You're not racing against anyone else. You're not behind. You're just on a different path. So after I finished my undergraduate degree, I decided to pursue a PhD at UC Santa Barbara, which is a very beautiful campus in Southern California. And I thought I wanted to pursue a PhD because I thought I wanted to teach at the collegiate level, and a PhD is usually a prerequisite for those positions. I also like the idea of being at the top of my field and earning a top credential. And I wanted to learn more about the research process. So at UC Santa Barbara, I worked under my advisor, Professor Song-Yi Han, pictured on the lower left there. And here's also a picture of some of my colleagues from also in the lab from my time there. And I was studying Alzheimer's disease under Professor Han's direction. Unfortunately, things didn't work quite the way I wanted them to, and I didn't end up advancing to candidacy. So I had a choice at that time. I could either try again to advance to candidacy, or I could leave with a master's degree. And at that point, I was feeling like I was ready to kind of leave academia, get out there, see what I could do in industry, and explore different career options. So with the help of my advisor and my lab mates, I was able to finish my master's degree, and eventually some of my research ended up in this publication shown here. So then I was unemployed and needed to find a job. And hopefully this is something that won't apply to many of you for a few years, but I wanted to give some advice because I've talked to a lot of undergraduates coming out, and they're always concerned that things aren't happening or happening fast enough for them, or that they're doing something wrong just because they don't have a job lined up for immediately when they graduate. So some of the best advice I received and that I thought was helpful was that you want to submit good applications, not perfect applications. When you're applying to jobs, you don't know what's usually happening internally in those companies. So you don't want to spend hours and hours and hours trying to get the perfect application together, only to send it to a job that's already might be earmarked for an internal candidate and is just being posted to kind of check a box. You still want to put out good applications, but eventually you just want to hit submit. You don't want to waste tons and tons of time trying to get it to be perfect just because you don't know what's going on internally at those companies. For that same reason, I advise people to apply through company websites. These are usually the most up-to-date list of what job openings companies have, as opposed to job site aggregators, things like LinkedIn or Monster. They're usually just not as up-to-date as the company websites themselves. I also recommend setting like a reasonable application goal. For myself, I try to do 2 applications per day, so I didn't feel overwhelmed, but still felt like I was accomplishing something each day. And the last thing I would advise is to just be kind to yourself. I was fortunate enough to have the financial flexibility that I could take my time looking for jobs, but it still took me over 9 months and over 130 applications and 3 job interviews before I finally got my first position. And that was a temporary position at REGENERON. I only had a 12-month contract. So after 12 months, the contract would terminate and I have to find a new job. And it was in REGENERON's formulations department, which is responsible for studying drug stability. But I accepted this job because I knew it would teach me a lot of important skills. I get to learn different instrumentation, different experimental designs. I get familiar with a lot of industrial processes, things like documentation, and that those skills were going to be valuable for when I was applying to my next job when that 12-month contract ended. And that's what I really recommend when you're thinking about accepting any job, especially if you have the financial flexibility to be patient, is ask yourself, what will you be learning at that job that's going to help you get the next one? Most people won't spend their whole career in a single job position. So you always want to be thinking about what are you learning, what skills are you developing, or what is this job allowing you, what skills this job is allowing you to display. That'll be helpful to build your resume and helpful to talk about in your next job interview. So using what I learned in that formulations job, I was eventually able to get a full-time position with Regeneron in the protein biochemistry department where I work now. So I worked in protein biochemistry for about 3 years, and I learned, again, a lot of different instrumentation, different experimental designs, and was first exposed to statistical tools that I found very interesting. Now, after 3 years there, so around 2021, I was beginning to think about, okay, well, what do I want to do next with my career? Where do I see myself going? And began to realize that maybe protein characterization, that wasn't necessarily what I wanted to do for the rest of my career. And I had become much more interested in these statistical tools. So in 2021, I decided to go back to school part-time and get another master's degree, this time in applied statistics. And I expect to finish that later this year in December. So I'm very excited to see kind of where my career goes next. For myself, I like the idea of not knowing what I'm going to be doing in 5 years. It excites me to know that my career could have many twists and turns ahead of me. And it's kind of that mystery of my career and science in general that I find intriguing. So this is a snapshot of Regeneron's clinical pipeline. And I just want to call your attention to, a lot of these are color-coded by kind of disease pathology. We have drugs that are in the clinic for cancer. We have drugs in the clinic for blood diseases, for general medicine, for infectious diseases, for inflammation diseases. So we have a very diverse clinical pipeline. And what's not shown here is all of the preclinical research we're doing. So stuff that hasn't even made it as far as the phase 1 clinical trials. On the right-hand side, I want to call your attention to the 9 approved or authorized drugs we have. And I want to highlight one, REGEN-COV, which I will be using later as kind of a case study to walk through because I think it's a nice case study that demonstrates a lot of the technology and science we do here. I should note and make a disclaimer that the 2 antibodies that are part of this cocktail, caricimab and imdevimab, were previously authorized for treatment and post-exposure prophylaxis of COVID-19 in the US and for treatment and or prevention of COVID-19 in other jurisdictions. But because data show that caricimab and imdevimab is unlikely to be active against Omicron variants, it is not currently authorized in any US region as of January 24th, 2022. Despite that, I still think it's an interesting case study to walk through and again demonstrates a lot of the science and technology we do here. But I just wanted to get that disclaimer out of the way. So here I have an overview of what the drug development process looks like. And you can kind of break it down into these 4 phases. We have a discovery phase, a preclinical development phase, and a clinical development phase, and then a regulatory and post-commercialization phase. So what happens during these different components is, for example, during target discovery in the earliest stage of the drug development process, what we're looking for here is handles or anything that's part of the disease pathology that we think we can hit with a drug that is going to change how disease pathology functions, either eliminate the disease or reduce the symptoms of that disease. And once we think we have an idea of something we can target, we start generating drug candidates against that target, things that bind to that target very well and bind only to that target, because you don't want your drugs that bind to things they're not supposed to bind to. Once we have one candidate or a handful of candidates that we think do this well, target well, we then move on to the preclinical development phase. Here we start looking at all the different qualities that those drug candidates have. We start doing our biophysical biochemical characterization. We start doing animal studies or cell-based studies to look at how well these drug candidates work. And once we establish that one or more of these drug candidates seem to be doing what we expect them to, then we move them into the clinical phase. Now, this is your typical phase 1, phase 2, phase 3. And as we move through those phases, early we look at just safety, and then later we start to look at efficacy, and we start these phased clinical trials start to go up in the number of patients enrolled in each one. So phase 3 trials are much larger than phase 1 trials. And again, once we have the clinical results, if everything goes well, we apply for what's called a BLA, a biological licensing application. And this is for the commercial approval, the approval from the FDA to finally begin selling our drug on the open market. And again, assuming that the FDA checks off all our work and gives us the okay, then we move into the kind of life cycle management post-approval. So there's a ton of different departments at Regeneron that have to come together to make this process happen. And I have a few highlighted here, but they're really too numerous to all fit onto the page. But I'll try to highlight just a few to give a sense of where they kind of fall in this development process and what their contributions are. So the Regeneron Genetics Center is involved in researching genetic components of diseases and trying to help us identify good targets for our drugs. We also have the Protein Expression Sciences group. This is the group that does the earliest first attempts at manufacturing our drugs at small scale, which leads directly into the preclinical manufacturing group, PMPD, which is responsible for taking the work from the Protein Expression Sciences group and scaling that up and developing robust manufacturing protocols. We also have our facilities department, which is responsible for keeping the lights on and keeping the building from falling down around us, which is very important for doing high quality research. And then we have like other departments like environmental health and safety, which makes sure we're properly disposing of all and any waste we generate, and also keeping our scientists safe as they do their jobs in the research labs. So now let's ask ourselves, what makes a good drug? Well, we can kind of boil that down to maybe these three qualities. We want our drugs to be efficacious or potent. That means that they work well, that they're safe, and that they're developable and manufacturable. So the way we evaluate potency and efficacy, the gold standard for this, of course, is the clinical results. If it doesn't work in the clinical results, it doesn't matter all the work we do prior. It really has to show that it works in people. So in our clinical results, we want high response rates. So that means we want a lot of different types of people that take our drug to respond to it. And we want large responses. So we want to see that our drugs have a big effect on whatever the disease is. But of course, clinical trials are very time consuming and they're very expensive to run. So we want to have a good idea that our drugs are actually doing the thing they're supposed to before we send them into the clinic. So we develop things that we can measure in the lab. So we have cell-based assays, and we have other types of assays, such as binding specificity assays. These check to make sure that the drug's binding well and binding only to the thing that they're supposed to bind to. For quality of safety, again, clinical results are the most important measure of whether a drug is safe. Again, we want it well tolerated by diverse populations. We want side effects to be as little as possible, as unserious as possible. But I also want to mention that we want the benefits to outweigh, really want the benefits to outweigh the risk. And what risk is tolerable really depends on the patient and also what disease is being discussed. You might imagine that a terminal cancer patient has a very different risk profile than someone who might be suffering from a quality of life disease that is debilitating, but not necessarily life-threatening. And again, clinical trials, they're expensive, they're time consuming. So we want to have some idea of how safe and how high quality our product is before sending it into the clinic. So we have a lot of different types of experiments we can run to measure different aspects of drug purity. And then finally, developability and manufacturability. So ideally, we want to produce a lot of drug very quickly at very low cost. And to a certain extent, we can do that, or we can try to at least move our manufacturing processes in the right direction. Also, we want our drugs to be stable and have long shelf lives. You can imagine it makes it a lot easier to supply our drugs to patients if they can sit on the shelf for 2 months and be stable, or sorry, 2 years and be stable versus something like 6 months. It just makes everything much easier to get our drugs and put them in the hands of the patients that need them. So how and where does biochemistry and biophysics contribute to these different assessments of drug quality? So here, highlighted again, pretty much everything outside of the clinic that we can measure on the benchtop in a lab is highlighted here. And I want to point out that drug purity relates to stability and shelf life through the fact that you can imagine our drugs are most pure when they come directly off kind of the manufacturing line. And then over time, as they sit in their vials, they can begin to degrade. So we need a measure of drug purity when they first come out of the manufacturing process, and then be able to check that drug purity as they sit on the shelf and have an idea of what aspects of drug purity are important for knowing that our drugs are still effective and safe for patients. So I'm going to start with this cell-based assay. Again, I'm going to be using REGEN-COV as an example model system to kind of walk us through these different experiments. So first, just a little background on how REGEN-COV works. As many of you have probably heard, the SARS-CoV-2 virus has something called a spike protein dotting its surface. And this spike protein is what allows it to bind to our healthy cells and inject its DNA load into the healthy cells and then begin its replication process. So the idea behind REGEN-COV is, what if we bind something to the spike protein to basically block it from allowing the virus to bind to our healthy cells? And to do this, we use antibodies, which we generally represent as these Y-shaped proteins, with the two top parts are binding arms, and those arms can bind to the spike protein. And we've done this successfully in the past with Ebola, where we developed a 3-antibody cocktail using very similar principles and technologies. So we had an idea that this could work for certain types of viruses. So in this case, we developed a cocktail of two antibodies, this casirivimab and imdevimab, and they both were designed to bind to different parts of the spike protein. The idea being that if you had mutations in the spike protein, as can happen in viruses, you would need to have mutations in multiple different parts of the spike protein to render all of your antibodies ineffective. I just want to reiterate that this cocktail approach lowers the risk of escape mutants being able to avoid your antibodies and then making your therapeutic ineffective, but based on the most recent emergency use authorization, REGEN-COV cannot currently be used in the US. So the way our neutralization experiment works is we take a pseudovirus and we package it with DNA of luciferase. And the important thing that luciferase does is luciferase gives off light. So the way this normally works is our pseudovirus will bind to our healthy cell model, inject that healthy cell with the luciferase DNA, and then the cell will begin producing the luciferase, which gives off light. So in the unmodified process, when this binding is happening and luciferase is being produced, we see our cells all begin to light up because they're producing this luciferase, as shown kind of in this very bottom panel here. The idea is once we add our neutralizing antibodies, they block the binding of this pseudovirus and block it from injecting the luciferase DNA. And then this stops this light production process. So you can see here, when we add our neutralization antibodies, we see darkness. And as we dilute down the antibody concentration, there's not enough antibody to block this process and the lights come back. So we can use this neutralization assay as a screening study. So what's shown on the right is the results of this neutralization assay. And the important thing to know is that the curves to the left are better at neutralization because they need less antibody to achieve the same neutralization amount. So we can identify the top three antibodies that are good at this neutralization. They're the 10989, 10987, and 10933. And 10933 and 10987 were the two that make it into the final cocktail, 10989 having a few unforeseen difficulties during the rest of the development cycle. So then we want to go on and assess drug purity as another measure of whether our drugs are going to be safe and effective. So there's many different things we can think of in terms of purity. So to start with, we're going to talk about assessing purity by size. So what I have here is representing a vial of REGEN-COV and formulated. And what's in that vial is something called formulated drug substance, or FDS. And what formulated drug substance is just a fancy way of saying the last thing to come off the manufacturing process right before it goes into the final vial. So this is what we can think of as the most pure our drug's going to be, because it's the thing that's coming directly out of the manufacturing process. So antibodies, these Y-shaped proteins, are about 150 kilodaltons in size when they're by themselves. So what we want to see, and what we run our experiment to do, is to see how much of our drug vial is this 150 kilodalton species. So in this experiment on the right, we have this blue trace, and larger things appear on the left and smaller things appear on the right. And what we see in this peak 4 is this 150 kilodalton species, which is the singular antibody, and that's about 98% of our drug vial. Now we do have some small impurities. So at around 1.9 milliliters, you can see a small hump labeled peak 3. And at about 2.2 milliliters, we have another small hump that you can see and labeled as peak 5. Now, these both represent impurities. So the question we might ask ourselves is, so normally our drug coming off our manufacturing process is 98% pure. But what if it comes off is 96% pure or 80% pure and has more of these impurities? What can we say about how much of a risk that is for our patients? So what we want to do as the protein biochemistry department is we want to take these impurities, we want to isolate them, and then concentrate them up so that we can actually measure them and then look at what the properties of those are. So when we do that for peak 3, we get this red curve here. And what we see is that the majority of that impurity that we isolated is this 300 kilodalton species, which is exactly 2 antibodies stuck together, what we call a dimer. So the majority of the impurity in our FDS, remember the stuff that comes off the manufacturing process, is this dimer impurity. Now we can also do that for the smaller stuff, and we can enrich up that peak 5 and see what that corresponds to. And we see it mostly corresponds to this 100 kilodalton species, which corresponds to an antibody with one of its arms cut off. And that's about 87% of that smaller impurity, what we call low molecular weight. And the bigger impurity is something we call high molecular weight. So now that we've figured out what those things are, now we want to look at, well, can we tell how effective they'll be at the neutralization process? Will they still do the thing they're supposed to? So we can go back to that neutralization assay and look at the relative effectiveness of the different impurities versus our original pure drug. So if we look at the FDS control, and we look at this all just qualitatively, we see that it has about a +3 effectiveness. And the enriched high molecular weight is about the same of effectiveness. So the enriched high molecular weight, this dimer impurity, doesn't pose much of a risk to our patients in terms of reducing the efficacy of the drug if we have a bit more of it or a bit less of it. Now, the enriched low molecular weight, however, is much worse at doing the neutralization. So we want to make sure when we're manufacturing our drug that we don't allow too much of this low molecular weight impurity, because it could impact the efficacy of our drugs when we finally give them to patients. Now, there's not just size purity that we can think about, but we can also think about assessing purity by charge. So what I'm showing here on the right is relative charge distribution. So these green zeros I have here aren't truly zero charge. It's just that they're, relatively speaking, the majority species in the vial, and that we think of charge based on everything relative to that species. But these are, in actuality, do have a certain charge. We just like to think of them as zero to make kind of it easier to assess where everything else falls. And what I really want to emphasize here is just because you have two things that are the same size does not mean that they have the same charge. So in this example, we have two singular antibodies, and we can see that they have very different charges. So knowing the size does not necessarily tell you what the charge is, and vice versa. Now, we tend to see that size does correlate with charge, and I think this will make more sense on the next slide as I walk through this. But it's not definitive. Knowing the charge or the size does not necessarily tell you what the other property is. So we want to do basically the same thing we did for size. So we look at our FDS control, that is the stuff that comes off the manufacturing process, and see that we have 7 different main charge variants. So what we want to do is isolate each of the 7 peaks and then look at it on that neutralization assay to see if it's still working properly. So this peak 5 is the main peak, and that's the majority species. And we can call everything to the left of it an acidic variant, and everything to the right of it a basic variant. So when we try to do this isolation and enrichment process, we'll get something like this fraction 1, where we try to isolate this first peak, this first species, and we're able to do that reasonably well. So we're able to get something that is 75% pure for that first peak. Now, we're not able to enrich it all the way. We still get some of the peak 2 contaminating our first fraction, but this is enough of an enrichment for us to get a good sense of how this peak 1 is behaving on our neutralization assay. And we want to try to do that for each of the other peaks, and we are able to do so to varying degrees of success. For example, we're only able to get that peak 2 enriched to about 50% purity. Fraction 3, we're able to get it to 70% pure for peak 3, and all the way down to the basics, which this peak 7 is so small in its amount, we're not able to enrich it as its own fraction, and it gets lumped in with this peak 6. So when we look at the neutralization assay, again, we're going to look at this qualitatively. I want to call your attention to 2 fractions. First, fraction 1 is the only one that performs slightly worse than the FDS control. And fraction 5 performs slightly better. So actually, if we could get rid of all the other charge variant species and only have fraction 5 available, this suggests that our efficacy would be even greater than it is currently. Now, that's, of course, much easier said than done. So going back to fraction 1, the one that we see has slightly worse efficacy, we ask, okay, is this being caused because of the charges that are in this fraction 1 in this peak 1? And the answer is not necessarily, because remember I said earlier that while it's not definitive for size, sometimes we get co-enrichment of size variants in with our charge variants. So in this case, this fraction 1 is actually 25% of the low molecular, contains 25% of the low molecular weight impurity. And as you recall from earlier, the low molecular weight impurity was the one that was least effective or not very effective at doing the neutralization. So in order to tell whether this effect of slightly reduced efficacy is due to the charge or due to the co-enrichment with this low molecular weight impurity, we would have to do more testing. So I want to end by going back to potency and efficacy and just showing some clinical results, because again, everything we do has to lead into being efficacious in the clinic. So here is some clinical results from an outpatient study. And there's a lot going on this page, so let's just walk through it. So in this first chart on the left, we have decreasing viral load over time. And as you can imagine, if you get infected with COVID-19, what happens is your body begins to fight off, produce an immune response and begin to fight off the disease. So that's what this gray line represents. This is the placebo given to people. So this is what we might consider the baseline kind of body response in absence of any intervention. So what we want to see is that when we give people our drug, that the body's capable of fighting off the virus faster and that their virus load goes down faster than it would in the absence of the drug. And that's what we see here in these kind of blue and purplish lines. And listed here in 2.4, this 2.4 gram and 8 gram is the different dosing. So we don't really see much difference here between the doses. So when we went to the larger study, the phase 3 study, which many more patients, we reduced the dose. And the reason we did that is because if you remember, there was a huge demand at the time for effective therapeutics. So if you can give people less drug and get the same effective result, that means you can stretch limited supplies to over a much greater population. So that's why we were able, and since we didn't see a difference in the first study, we were able to change that to look and see if we could lower that dosing for the second study. Again, I just want to reiterate that this study was conducted on previous variants of the COVID-19 virus, and that REGEN-COV is not currently authorized for use in the US. So just to kind of summarize what we've talked about what makes a good drug, hopefully I've done a good job of convincing you that lab-based biochemistry and biophysics type experiments can inform us about what we might expect from clinical results and are an important part of the drug development process. And then finally, I want to end with my pitch for STEM education. STEM degrees are very versatile, and there are many roles in pharma suitable for STEM, but also I want to say that to work in pharmaceuticals, you don't necessarily have to have a STEM degree. We have a lot of people who work and are part of the team that make the drug development process happen that do not have STEM backgrounds. And finally, I want to tell you to pursue your passions. And just remember that careers are long and can take unexpected turns. It's important to be patient with yourself, confident in your decisions, flexible towards change, and resilient in your careers. And now I can take any questions.